odd sight to see from the guy who's not a monk. See that? That's definitely odd.
There's a problem with this scan. Go away.
They let the outsiders, the people that weren't in the monastery, into the church.
They can't come in the monastery. We have a couple people who worked in the monastery, but that's about it.
Gotcha. So that's what I was thinking. If there weren't any outside people that are normally in the church, then it's just you guys. I don't see why you have to wear a mask very often, but hey,
Well,
stay cautious, right?
well, one of the things is, the main thing is that the fact that now we're interacting with you guys every day.
Yeah, now you're back. Yeah, you're in here. The rooms, in the cafeteria, always.
Allison, how are you? Recording. Okay, cool. Okay. Woo, 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 woo.
Well, that's everyone out today.
No.
I told him plastic candle.
You told Emily class. I know where Emily is. Emily Rome, but.
King of the Father, Son, and Holy Spirit.
God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. Father, Son. All right, by way of reminder, quiz 1 is still available. It will be available until 6 o'clock tomorrow. So that's your last reminder. You are all in your classes. You can handle this. Allison, can you hear me?
Yes.
Okay, good. Good. All right. Review. So, action potential, right? We initiate an action potential somewhere near the cell body of a neuron. And what kind of a signal is this? Is this electrical or is this chemical? It's an electrical signal. It has 3 parts. Right? Depolarization, and depolarization always means the membrane potential is becoming more positive. Then we have, and then during depolarization, sodium is coming into the neuron. So we're only talking about the movement of these 2 ions, sodium and potassium. Then during repolarization, the inside of the neuron is becoming more negative. That's because what is moving where? Emily.
The potassium is moving inside the cell and sodium is moving outside the cell. I mean,
Which one should move out?
potassium moves out because of time level and sodium.
Right. So sodium moves in during depolarization, potassium moves out during repolarization. Then we have this concept called after hyperpolarization. This is really important because hyperpolarization means that the membrane potential is becoming more negative in relation to resting membrane potential. We're going to need to know these two terms, depolarization and hyperpolarization. We're going to see how those are important for generating an action potential today. But before we do that, once we initiate an action potential somewhere around here, the axon hillock, close to the cell body. Once we initiate an action potential here, this action potential gets regenerated, boing, 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 at each point down this structure, which is called the axon, right? And that's called propagation. So when this action potential moves down the axon, we call that propagation. And the way this happens is by a phenomenon known as local current flow. So here's basically what happens. Now, what are these blue things up here? These blue things are, okay. I can be really goofy because nothing is filming me right now. So I can turn around. These blue structures, they are voltage gated sodium channels. All right. Bertie and Devin have had these concepts before in depth, in detail. So I'm going to ask the psychology side of the room, that includes you out there in video land. You don't like your face out there. We caught that. Anyway, so when you hear this term, voltage gated sodium channels, what's that term mean to you? If you were to describe that to a classmate, how would you describe that? Based on what we talked about Thursday. Okay, Emily. Good.
From the voltage, we know that it's electrical, so we need electrical current in order to open up those gated
Good.
channels with sodium.
Okay, so we need an electrical current. And what tells you that it needs to open up? Good. If it's gated, that means most of the time it's closed. And sodium tells me
That's what the sodium goes through the channel.
that's what can move through the channel once it's open. That's awesome. Awesome. Good, good, good. All right. So these are four voltage gated sodium channels, and they are, I'm showing you, I drew them as being closed. They're closed. Resting. So what happens then is that when the action potential happens, so here, so when you think action potential happening, that means that the membrane potential has become more positive to reach the threshold potential. So in this case, it has to increase from -70 to -50 millivolts in order for that channel to open. That's threshold. So when you say the neuron reaches threshold, that's what it means. You get an action potential. Okay. You guys with me? Are you with me out there?
Yes.
Good. All right. So the channel opens. Channel opens. Notice the change in shape. And now sodium, we're going to say, rushes into the neuron, right? And we call this sodium influx, correct? Correct. We call it sodium influx. All right. That's depolarization. So if we're scoring this at home, that's this part of the action potential, the rising phase. And it's happening, we're saying sodium is coming in right here. Okay, now, right here comes my, here it moves. Wasn't that awesome? Let me do that again. Ready, Allison? Watch it, watch. Hey, quick, ha! It just moved into the neuron. You see that? You didn't see that, that's okay.
Yeah, I did.
So, sodium is now inside these cells, all right? That's what I'm showing you here. Now what happens, okay? If you took diffusion, are we familiar with that term, diffusion? Heard diffusion before? So, you take some food color, you drop it in water, drop, drop, drop. We'll put some red food color in, drop, drop, drop. Does it stay in one place? If you put it in the water, what's it do? Spreads out. That's called diffusion. So things moving from high to low concentration, if it's moving through a channel, that's kind of like controlled diffusion, right? That's controlled diffusion. But then once the sodium comes into the cell, it's got room to move, man. It can move everywhere. So what these arrows are showing then is, well, first of all, the little pluses, these pluses are representing sodium that's moved into the cell. And then the blue arrows are showing that, well, it's real concentrated here initially, then it starts to move away or diffuse. Okay, diffuses away. And so now, as these ions move away, let's say they move down to this receptor to the right, which is now closed. If I dump a bunch of positive charges right here, what's going to happen to the membrane potential here at this receptor? Is it going to change? Is it going to become more negative? Is it going to become more positive? Yeah.
It's going to be more positive.
It's going to be more, I'm adding positive charges right here, right? So the sodium comes in here, positively charged, diffuses, moves away from where it comes in. Now it's making this region more positive. So there's my sodium moving away, whoops. And when it diffuses away from this channel, what happens is increases, right? When it does this, the membrane potential we're going to say at the next sodium channel becomes more positive. Because there's now sodium there that wasn't there before. As a matter of fact, I'm going to say that it raises the membrane potential, MP, at the next sodium channel to threshold. So it actually is going to make the membrane potential here, it's going to increase it to threshold. And when that happens, what happens now? What do I now generate at this sodium channel?
Is it like a higher concentration that you have of sodium?
Well, when I raise the membrane potential to threshold, what do I get? Another action potential. I get another action potential. So that means that this sodium channel then opens. Sodium comes in here at the second sodium channel, diffuses away, raises the membrane potential at the next sodium channel to threshold. I get an action potential here, then here, then here, then here. That's how this signal moves. So I can get an action potential here, and then here, and then here, and then here. At each point on the axon until that action potential ultimately reaches the axon terminal.
I have a
Right.
question.
How the signal moves. Yep.
I'm just trying to read your handwriting. After the second arrow, it says raises membrane potential, and is that a negative sign? Okay.
At next sodium channel to threshold, I get an action potential at the next sodium channel.
And I have a question too. Hello?
Go for it.
Oh, okay. So this is essentially the process that we talked about last class when the action potential arrives to the axon terminal and it activates the calcium channels. So this is essentially like taking a step back before that process and talking about what generates an action potential.
You got it.
Okay.
So yeah, what we did was we jumped ahead and then we reversed.
Okay, that makes sense.
Now we're moving from the cell body. And so basically, Allison, this process that we're going over right now keeps repeating from the cell body all the way to the axon terminal.
So this happens prior to the release of the neurotransmitter from the synaptic vesicle.
Absolutely.
Okay.
But the initial action potential, this is a good question. The initial action potential that's generated in a neuron is typically generated somewhere around where that region called the axon hillock, which is close to the cell body. That should be labeled. Is that labeled in your notes?
Yes.
So that's where the first or initial action potential in the neuron, is generated. Then it gets propagated, which means it gets regenerated and moves down the axon till it reaches the axon terminal. Does that get you?
Yes. Yeah, that makes sense.
That's a great question. Okay. Awesome. Any other questions about that, Julie?
So as you're going down the axon and you're creating all these new action potentials,
Hmm?
do all the action potentials get down, or do they kind of die off as they like?
Another great question. So every time you generate an action potential, did you hear the question, Allison?
Can you repeat it one more time? I was writing.
That's okay. The question is, as the action potential, so the initial one is initiated here at the axon hillock, and then it's regenerated as on each point down the axon.
Mm-hmm.
The question is, does it diminish Or does it sort of fade away as it goes? Kind of like, yeah.
Yeah.
No. The action potential is always the same amplitude. It's always going to increase the membrane potential somewhere around 100 millivolts. It's always going to look like this. There are events that change the membrane potential that do diminish over distance. And we're going to talk about those today. Okay, Julie.
But is there like a bunch of like action potential, there's just one that's kind of like,
Oh, it gets repeated. So,
okay.
All right. So if we were to draw this, let's see here. Well, you guys can explain this to Alice later. All right. Okay. So we get the first one here. Looks like that. Let's say it moves down here. It's going to be the same. Each time it's generated, it should look exactly the same. Trying to draw them the same, I'm not a very good artist. So until we get to the here, it's still the same, and then finally, we're going to get the release of the neurotransmitter. Always the same, always the same. Because we have sodium channels at each point. So it's the opening of the sodium when these sodium channels open, the sodium rushes in. So you're always going to get this big, huge influx of sodium at every point where we have sodium channels. All right. Does that get you? Okay, cool. So it gets regenerated. Choo, choo, choo. If there was just one, if there was only one, and the sodium had to diffuse all the way to the end of the axon terminal, there's no way that would happen. Let me give you an example. We're talking about maybe little neurons that are in your, stretching from one region of the brain to the next. We have a sensory neuron in your right, let's say your right big toe, so the sensory receptor is in your right big toe, so move your right big toe. That neuron has axons that go all the way up your right leg, enters your spinal cord, goes all the way up to your brainstem. That's one cell. There's no way there's enough sodium to travel all the way to the end of that cell to get sodium, to get the neurotransmitter to release. So because of that, then the action potential, it gets repeated. It's always the same. Yeah. Good. Good question. Good question. So are we cool? All right. Awesome. So just as depolarization, the rising phase of an action potential, happens at every point on an axonal neuron where there are sodium channels, repolarization is also happening. So immediately, once the sodium diffuses away at this point, sodium channel closes, then you're going to have potassium channels open at this region, and potassium leaves. That contributes to the falling phase or the repolarization. So that both of those steps are happening. Both of these steps are happening. Actually, and even hyperpolarization. These steps are happening at each point in the axon. Okay. Just to keep that in mind. All right. And here again. Okay. So, we've already talked about this, about how at at least in this case, in these settings, we're only talking about the movement of two ions here. Sodium can move in, potassium can move out. We said that the inside of the neurons become more negative due to potassium efflux. Okay, we've said that. And this is something that we want. This is a concept that we're definitely going to work on a little bit in class at some point, not today, but at some point, maybe next week or a couple weeks, when we start talking about, actually, we might do it on Thursday, excuse me, how the movement of different ions across this plasma membrane can contribute to the membrane potential becoming more positive or more negative. That's important, especially when we start talking about what happens in the postsynaptic neuron. Whether or not that neuron, there's a greater likelihood of that neuron generating an action potential or not. Okay. So now, myelin. Who's heard of myelin before? Okay, so what is myelin? Derek, please.
Is the wrapping around of the nerve insulating it?
Okay, so there are cells that are very high in fat content that wrap, actually, this is sort of a cross section. These would be layers around neurons. That's called myelin. So it's insulating. The other thing it does is that in neurons that have myelin, not all neurons have myelin. Some are unmyelinated. But in neurons that have myelin, we only have sodium channels in these little gaps. So the cell wraps around, let's say here we've got 1, 2, 3 cells that are wrapping around these neurons, and then there are regions where the membrane, the plasma membrane isn't covered by myelin. Those are called the nodes of Ranvier. Nodes of Ranvier. And in neurons that have myelin, the sodium and the potassium channels will only be found in these nodes of Ranvier. So then you only have action potentials that would happen here, and then here, and then here. And so then it sort of has the look, or it appears as though the action potential is jumping from node to node. So, and that's called saltatory conduction. Why do you think it's called saltatory conduction? Okay, there.
It's very factual on here, yeah. And I don't know the proper English, but like whenever you do like a somersault, it's like somersault from node to node.
Okay.
That's where it comes, because like in the Hungarian translation, that's not what it is.
Saltatory. I've always thought it was because the sodium is moving in.
That's the other explanation I know. But if you do that, we go with Hungarian.
So it's only moving in certain regions.
Yeah.
Okay. So what happens then is the sodium, let's say we have an action potential here. Sodium represented here in green moves in. It diffuses away. And there's enough sodium to come in, but when it diffuses away from this point of entry down to the next node of Ranvier, it's able to generate an action potential here. Okay. So, myelination. Not all neurons are myelinated. what do you think happens to conduction velocity? So conduction velocity, that's the speed at which the action potential moves from one end of the neuron to the next. Would you think that myelinated, yep, okay. Would you think that conduction velocity would be faster in myelinated or unmyelinated neurons? It's going to be faster in myelinated neurons because this process, even though it's fast, it still takes time for channels to open, cilia to come in.
I have a question.
You have to generate action potentials along the entire length of the neuron. Now you have a very slow velocity. Allison?
I thought that the myelin was just located on the axon itself.
It is.
Oh, okay. I thought you said that it was on like the cell body, like it was on the entire neuron.
No, it's just on the axon.
Okay.
Oh, so myelin cells. The myelin is actually a living cell. So it's not just tissue. It's a living cell, and so that wraps around the axon. Maybe I did say cell body. I may have. So. Okay. Are we good?
Yep.
Okay. And the other factor that affects conduction velocity is the diameter of the neuron. Real easy. The larger the neuron, the faster the conduction velocity. You just got more stuff going on. Got a bit more bandwidth. Now, who can think of some, he can maybe name some diseases that affect myelin? Multiple sclerosis. Yes, I do believe so. So, some degenerative diseases, the myelin cells get attacked. And now, and then those neurons just become very, very, very inefficient at conducting action potentials. Really impairs your movement. Okay. All right. Let's stop there for a second. Questions so far? All right. Yep, John.
If you're looking at like axons from a microscope, how can you tell that they're myelinated from that? Like, would it show something different or?
Yeah, you'd be able to see. The main way you would do it would be to look at an unmyelinated versus a myelinated neuron, and you would be able to distinguish that. Electron microscopy. You'd have to look at very high magnification. Okay, now, ultimately, the whole point of talking about all of this is action potential generation, what makes the membrane potential has to reach threshold, how can the membrane potential become more positive, how can it become more negative. Ultimately, these are the things that we want to talk about when we start and consider when we start thinking about what happens at a synapse, right? Because a neuron might make how many, like 1,000 or more connections with other neurons. So there's going to be a lot of input to these neurons that are affecting the membrane potential. So what's going on here? So here we're going to, this little setup, this is taken from your book. At the stimulator, so here's our pyramidal neuron. You might recognize this. These are all our, what structures are these little spiny things? These are dendrites. So we're infusing or inserting, a little electrode into the cell body. And then we're recording, we're going to measure the membrane potential here. And everything we're going to talk about is in relation to this resting membrane potential. So this dashed line right here, it separates the blue from the green. That's our resting membrane potential. There's lots of stimuli that can open, or that can open ion channels momentarily. So there are many different kinds of stimuli that can open ion channels momentarily. Momentarily, leading to brief, localized change in the membrane potential. So there's a lot of different things. Stimuli.
Use that all again.
Yes.
Understand, but thank you.
Absolutely. So the various stimuli that can impact the neuron and that can lead to the opening of ion channels momentarily, that can lead to a brief local change in the membrane potential of the neuron that's affected. And we are going to talk about these in terms of whether they are depolarizing or hyperpolarizing. Now, we've already seen these terms, right? Before we do this, I want to get my eraser. I want to introduce another ion to you. Start introducing some of our other friends into this. So here's the membrane, here's my plasma membrane. We've already said, and drew this dashed line, we've already said that the concentration of sodium is high outside, right, and it's low inside. We've also said that potassium is high on the inside and low on the outside. By the way, I know some of you folks have heard this before, but a good mnemonic to remember where sodium is high and where it's low, and where potassium is high and where it's low, is the old salty banana mnemonic. Take a banana with its peel on. Pour a bunch of salt on the outside of the peel. What's salt made out of? Sodium and chloride. So how much of that salt is actually getting through the peel? None. So sodium is high on the outside, low on the inside. What's one of the main things in bananas? Why do you eat bananas? Potassium. Bananas on the inside of the peel. Potassium's high on the inside, low on the outside. But since salt is also chloride, I need to introduce our little friend, chloride. Because chloride is also high outside of cells and low inside cells. All right, so allicin chloride, Cl minus, is high outside of cells and low inside of cells. So, we said depolarization. This is when the membrane potential becomes more, I can't hear you.
Depolarization,
Yep,
something more positive.
there we go. Praise God. Hallelujah. Can I hear the church? So, looking at these three ions, sodium chloride and potassium. If we have an event, some sort of event, stimulus, so let's use the sense of touch. If you can feel it, well, anytime you touch your skin, it's possible to touch your skin without being able to feel it. But if you activate sensory receptors, the inside of that sensory neuron is going to become more positive. It's going to be depolarized right where the sensory receptor is. So which of these three ions is probably moving what direction to make the inside more positive? First of all, what direction can sodium move? What?
High or
So in or out?
low?
Good. What direction can chloride move? Jonah?
In.
Good. And then we know that potassium can move out. So, depolarization, movement of which ion, which way? Devin?
Sodium in.
Sodium in. Probably what's happening. Sodium influx. All right. Hyperpolarization. Hyperpolarization is when the membrane potential becomes more negative than the resting membrane potential. Okay. You apply stimulus, becomes more negative. What would be some potential ion movements here that can lead to a hyperpolarization? How about give me one? Julie? Chloride in, no. Yes, we open chloride channels and that chloride comes into the cell. It carries a negative charge, so it's going to bring negative. Okay, what's the other one that we've talked about a lot before? Devin?
Potassium out.
Potassium out. Okay. Potassium efflux. All right. Awesome. So somehow we're injecting current A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P. Do they have that number?
Well, we have a longer alphabet, we have 43 letters.
You have 43 letters. So your songs, is it the same? It's a different melody.
No, I don't even think we have a song. So for us, S and C together, that's one letter. C and S together in the other order, that's one letter. C and S, one letter. We have two types of O's. We have, technically, I guess we would call it four types of O's because we have O, we have like two versions for every.
O, and
Yeah, I can hear right now if you want, but I don't know to speak.
Okay, sorry. So, anyways. So let's take a look here. We apply, we inject some current into this neuron. What's A? Hyper or dipole? What's A? Emily?
Hyper.
Hyper. Hyperpolarization. you likely to get an action potential more or less likely to get an action potential here? Less. What about B? What about B, John? What about B? Hyper. Is B a bit, that's actually more hyper than, that's like super hyper compared to A, right? Okay, good. So we can say maybe chloride's coming in or potassium's going out here. Maybe both chloride's coming in and potassium's going out. We don't know. Okay, good. What about C? C, did we get an action potential with C? Okay, first of all, is it D or hyper? D. It is depolarization, but not enough to generate an action potential at the next neuron. So, this is important. You can, it's possible, having input from, let's say, 100 different neurons to a neuron, and the result could be that the membrane potential is more positive, but not enough to generate another action potential. So that, whatever that signal is, whatever that stimulus was, whatever that thought was, you know, maybe that's when you go, you know, I can't remember. Right there. You didn't hear that. Right there. That's what I did. Okay, there. Bang, it's recorded. Okay, good. What about D? What about D? What about D? Bam! Well, actually tells me you got action potentials, right? So here, these little spikes, these are each action potentials. So for D, E, and F, the signal keeps on moving on to the next area. All right? Yeah, Emily.
On the graph that's labeled local potentials, what does that like mean in comparison to action potentials?
Great question. This gets back to Julie's question. So local potential. All right. Did you hear the question, Allison?
I did a little bit.
Okay. So the question is, on the graph, Emily said there are some that are labeled local potentials, and then there are some that are labeled action potentials. What's the difference? Okay? So the local potentials are going to be changes in the membrane potential right where this microelectrode is measuring. And if we were to measure, let's say, I'm moving down the screen now, down on this axon. If I were to move halfway down this axon and measure something, if I had another recording electrode here, for example, would I who thinks that with A, B, or C, that I would actually record something down here? What do you think, Allison? If I had a recording electrode about halfway down, let me go over to my screen here. I'm going to make a circle. Yeah. Pen, like right here. It's really, see, if I, hey, okay. So if I had a recording electrode, another recording electrode, where the red circle is, would and if we're recording following A, B, or C, would I be able to detect any change in the membrane potential down here? Who says yes?
I don't I would say no.
You say no. I say no. You are right. That's because A, B, and C are local potentials. They're only affecting the cell. We're going to say really, it's local. It doesn't travel outside of where the change in the membrane potential actually happens. Now, action potentials. So, local potentials, they happen at the site where the stimulus happens or when the input happens, but then they diminish as it moves away from the site where they're initiated. They're not regenerated. What about action potentials? So I get an action potential where the electrode is actually in the cell body. Let's take a look at D, for example. Would I then generate, or would I then be able to record change in membrane potential where the red circle is? Yes, because an action potential is regenerated at each point along the axon once it's initiated. Got that? Good, that's a good question.
I have another question.
Go for it.
So when hyperpolarization is occurring, there can't be
Yep.
an action potential present.
Uh-huh. Oh, they're
The only
Say
time
that
you
again?
have an action potential occurs when depolarization is taking place.
That's correct. An action potential will only happen, well, we're going to see something happen on the next screen. So an action potential will only happen if the membrane potential is increased, made more positive to reach threshold. So this, what we're looking at here, is a very simple model looking at one neuron following a single stimulus. That's not what happens in the brain, right? Normally, a neuron is receiving input from hundreds or possibly thousands of other cells. So let's take your question, Allison, and let's go to the next screen. Well, maybe not quite yet, all right? The next screen, the next screen. Okay, Julie.
On that last screen, they're on, say, like D, after it hyperpolarizes, and then it looks like it goes back and, like, it gets that threshold line. Why does it go back down?
It's just this is the neuron establishing equilibrium. Just the movement of different, some ions in, some ions out. It's just sort of, can also be the result of the remnants of the electrical stimulus going through. There's lots of things that could be happening there. So, good question. Yep. Okay. Now we're going to take these terms, depolarization, I flipped them in, hyperpolarization, and we're going to talk about them in terms of EPSPs and IPSPs because we want to start talking about what happens in networks of neurons, right? That's what we really want to talk about. What's going to happen in networks of neurons? And we're not going to talk about real complicated circuits, but we're going to talk about some things. So, IPSP. IPSP. Well, in order to talk about this, we have to have two neurons. So let's, okay. So we have on the screen here, so we have neuron A, right? This is called the presynaptic neuron. So neuron A is the presynaptic neuron. Neuron B is the tan one, the beige neuron. This is called the postsynaptic neuron. Okay, so we've talked about these before. first of all, what does IP, oh, if you've done the reading, what does IPSP stand for? Inhibitory post-synaptic potential. How do we get an inhibitory postsynaptic potential? So here we're talking about a change in the membrane potential in neuron B. And if we're talking about hyper or depolarization, which of those two is it likely going to be? Hyper or depolarization? Inhibitory. I'm inhibiting, I'm reducing the likelihood of an action potential in neuron B. Okay, so, and this postsynaptic potential is the result of the interaction between neurotransmitter and its receptors. So an IPSP, this is when the neurotransmitter receptor interaction results in hyperpolarization. It's less likely that the postsynaptic neuron is going to generate an action potential. So then EPSP is excitatory postsynaptic potential. PSYNPOT, postsynaptic potential. This is when a neurotransmitter receptor interaction results in depolarization.
Is it presynaptic or postsynaptic?
Post, yep. So nerve receptor interaction, IXN, results in depolarization. So you will hear about, there are neurotransmitters that are simply inhibitory. They are inhibitory. The big one is GABA. GABA, DABA-do. And Fred Flintstone, you don't even know who Fred Flintstone is. So GABA. You know who Fred Flintstone is?
Yeah.
GABA, DABA-do. All right. So GABA is the main inhibitory neurotransmitter. When it binds and activates its receptor, inhibits the neuron, the postsynaptic neuron.
It's GABA.
GABA.
Okay, isn't it hyphenated with another A too, or is it just GABA?
GABA. GABA A is a kind of receptor. GABA, GABA, you are the dancing queen. No, that's ABBA. So anyways, all right. And the main excitatory, there's two. Glutamate and acetylcholine. These are the main excitatory neurotransmitters, which means when they bind to their receptor, always going to result in that postsynaptic neuron being more active. The other ones that we've talked about, dopamine, serotonin, norepinephrine, depends on what receptor the neurotransmitter binds to. Sometimes it's excitatory, sometimes it's inhibitory. Okay? So what that tells me then, well, that this is really important. It's the neurotransmitter receptor interaction that's going to then lead to either an IPSP or an EPSP. All right. So in review, neuron A releases its neurotransmitter, binds to receptors on neuron B, and the membrane potential becomes more negative. That is a what? IP or EP? IP, right? And if the neurotransmitter binds to its receptor, and the membrane potential becomes more negative, or more positive, then it's an excitatory, okay? Now, we're also going to, I'm not going to talk about that yet. So, cool. All right. So, in the brain, all right, in neurons, we talked about how in the cell body, integration is one of the things that happens, right? And we said that integration of all the EPSPs and all the IPSPs, we can have inhibitory, excitatory neurons. We can have IPSPs and EPSPs occurring in the same neuron, and they do. And then whether or not, and they are called, they're summated. There's sort of a net signal that happens. Is there going to be an action potential here or not? That's the question that's asked. And so, if the summation of all the IPSPs and EPSPs raises the membrane potential to threshold, makes it sufficiently more positive. So if that summation of all of those inputs raises the membrane potential to threshold, then you get an action potential at the axon hillock of the postsynaptic neuron.
Can you say that again, please?
Yes. If the summation of IPSPs and EPSPs on a neuron, and we're going to assume, for example, that these two reds and these two blues are 4 different neurons. And this is still a simple circuit, right? Remember, this thing could be receiving input from 1,000 other neurons. So if the summation of all of those raises the membrane potential to threshold, makes it more positive to reach threshold, then an action potential is generated in this postsynaptic neuron.
Thank you.
Yes. That's a good question. And it's also important to know that these IPSPs and EPSPs, that their effect on the membrane potential is very short-lived. So it's not like it's releasing the neurotransmitter and it's sodium's coming in, coming in, coming in, coming in, coming in, still coming in, still coming in. No, it's like boom, 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 milliseconds, boom, boom. On, off, on, off. Very brief. So, let's take a look. The example to the left. Reds are EPSPs. Blues are IPSPs. We're going to assume this all happens at the same time. Shazam! So, which of these two is more likely to generate, which of these two summation inputs, we'll put it that way, is more likely to generate an action potential? The one on the left or the one on the right? And why? Emily, you're quick. Okay, go ahead.
It would be the one on the right because it has the ESPs and the IPSPs, which is the depolarization and the repolarization, so that it would be able to do like the action potentials quicker.
Okay. Now, normally in an action potential, we do that. We depolarize, we repolarize. Are these action potentials or are these those local changes? Where does the action potential happen? Right here. So what we're looking at then is we're looking at, let's go back here for a second. Okay. So I'm glad we're talking about this. The two reds, they're only going to, okay, so EPSPs, if I have two neurons releasing neurotransmitters that result in the membrane potential becoming more positive. So first of all, is that a depot, are these, so those are EPSPs, the membrane potential is becoming more positive. So in this case, is that membrane potential getting closer to threshold or further away from threshold? Okay, so let's, closer to threshold, all right. Erase our ions here. Okay, so let's do this. Here's my resting. Here's my threshold. Okay. So we're starting here, and then the one on the left. So we got like a bump and a bump. Let's call it that. Actually, I shouldn't. That's not really correct because they're happening at the same time. So we'll do like a little bloop and a little bloop. Same time. The one on the right, what's going on? I have two EPSPs that make it more positive, and then what else is happening?
Two ISPs making it negative.
So the one on the right may actually, there may be no change in the membrane potential here at the axon hillock. So we could say that IPSPs kind of cancel EPSPs out in a very simple world. Okay? So then, it may be more likely that we would see an action potential generated in the left side than the right side. All right, Jonna,
What's up?
you got that?
Yeah.
Okay, cool. Allison, you with us?
Yes.
Good. Good. So, okay, so that's good, Emily. That's good. Are you? Is that cool?
Yeah, that makes sense.
But that's the difference. That's the difference. That gets back to your question about what's the difference between here and here. IPSPs and EPSPs, that's all affecting right here. So what we're asking is, do these inputs, does this summation of all of these inputs increase the membrane potential and bring it to threshold or not? All right. Well, this is the normal synapse that we looked at, that we typically look at, right? We typically discuss maybe in other classes where you have presynaptic neuron, has its axon terminal, it releases it on the cell body of a postsynaptic neuron. Synapses are actually a little bit more complicated than that. All right, they're a little bit more complicated than that. So let's talk about these three. And these words, the first word is about, this is where the axon terminus, so axo is always in relation to the presynaptic neuron. So the prefix axo is in relation to presynaptic neuron. The second part refers to the region where the postsynaptic neuron, where the synapse happens. So the first one, axodendritic, all right, is a synapse that connects an axon with the dendrite of another neuron. That's axodendritic. So it connects the axon terminal of one axon with the dendrite of another axon. That's the more classic synapse that we usually talk about. Axodendritic. Axosomatic is when a synapse is formed between an axon of one cell, one neuron, and the soma, the cell body of another one. So this is also pretty normal. Pretty normal. I don't have any text right now. All right, this is pretty normal. Presynaptic neuron binding the postsynaptic neuron. This is very, shall we say, this is normal. The third one, this is the weird one, axonic. So axodendritic and axosomatic synapses, what are they going to do? What are they going to do? Wow. What are they going to do? They're going to impact that summation. They're going to affect whether or not the postsynaptic neuron generates an action potential. That's always what we talk about. Now we're going to talk about a new wrinkle. We're not going to really, I'm just introducing this to you so that way you know that there are different kinds of synapses. Axonic. Axon terminus terminates near the axon terminus of another neuron. What? What? Why would you do that?
Say that again, it terminates itself?
Nope.
Oh, okay.
So A and B, we've got two neurons. C, we got a third neuron coming in here. So this one starting down here in the lower right, we've got our axon coming out. It's going to release. So if we look at letter C, we're looking at C now, Allison. All right, you with us? Letter C. Letter C. Letter C. So the axon at the lower right corner It's extending its axon and it's terminating on the neuron at the top of the little block here. So let's focus on this synapse first. This is an axosomatic synapse, right? Now we bring in the third one coming in from the bottom left. Axonic synapse. Why would you be releasing neurotransmitters onto the or near the axon terminus of another neuron? What's the point of that? Think about that for just a second. Let that sink in. Just see, these things are a little bit more complex than we make them up. Okay, Bertie?
Because you can cause action potential promotion of access rapidly, and you can inhibit at the end of the action. There's no one neural action, you can destroy that.
Okay. So you can basically increase or decrease the likelihood of neurotransmitters being released at this other axon terminal. It can either shut it down, or it can either stop the axon terminal, or it may diminish it, or it could, yeah. So this axonic synapse then can either modify the amount of neurotransmitter that's released. This is actually one of the effects that nicotine has. Nicotine, and we've heard of the region called the nucleus accumbens. We've talked about that. But one of the things that nicotine does is it binds to nicotinic receptors that are on the axon terminal of a dopamine neuron in the nucleus accumbens, increasing dopamine release. Which means that there's also, since nicotinic receptors are their receptors for acetylcholine, that means that there are acetylcholine neurons that release their neurotransmitter onto axon terminals of dopamine neurons, increasing the release. So that's just to give you an example of something that's a little bit outside of the box. All right. Don't forget about the quiz. We are going to dive into synapses and neurotransmitters, and we're going to start talking about the synapse. So, to definitely be finished with the reading for the new. There's going to be lots of new little reading outlines that pop up in your Schoology site. But we're definitely going to cover the synapse and the rest of this section on Thursday. Don't forget about the quiz. Quiz. Complete it.
Can we answer questions about the quiz on Thursday, once everybody finishes it?
Absolutely.
Okay.
Don't forget to clean your desk. Yes.
Father Sean, I like your sound effects. Another drug control.
Oh, I didn't say that today.
Must not get drug free there.
Oh, wait a minute. It is.
Thank you.
See you later.
Have a good day, Father.
See you, Jonna. Devin.
Talking about the Hungarian alphabet,
40 letters?
43 I believe, but the issue is that